look at your hands for a second. Like, really, look at them.
Yeah, just hold them up.
Feel the skin. Think about the muscles working underneath, the incredibly complex DNA coiled up tightly inside every single cell making up those fingers.
You feel solid, right?
Yeah, you feel self-contained. But the truth is, the very building blocks making up those hands, your DNA, your proteins, they're the result of an invisible, microscopic war.
A very old war.
Exactly. One that has been raging across the planet for, well, billions of years.
And it's a biological war over a gas that is, somewhat ironically, literally floating right in front of your face as you breathe.
Right. And to figure out how that war was won, we are doing a deep dive today into a comprehensive Wikipedia article on nitrogen fixation.
It's a fascinating topic.
It really is. Our mission for this deep dive is to track how an incredibly stubborn, invisible molecule floating in the air gets transformed into the literal foundation of your existence.
The stuff that makes you...
Yeah. We're going to explore how nature struggled for eons to pull this off, and how a human-engineered shortcut to bypass nature's limits completely exploded the global population.
It is arguably the most important chemical process on Earth, and yet it happens almost entirely out of sight.
Right. Because when we think of things we need to survive from the air, we automatically think of oxygen.
Oh, always. Oxygen gets all the glory.
Exactly. But nitrogen is this hidden architect. We absolutely need it to live, and we are completely surrounded by it. I mean, the atmosphere is roughly 78% nitrogen gas.
78%. It's everywhere.
But for the vast, vast majority of life on Earth, we are completely incapable of using it in that form.
Yeah, it's a classic paradox of plenty. To understand why we can't just, breathe it in and use it to build our bodies, we have to look at the underlying chemistry.
Right.
Atmospheric nitrogen, which is called dinitrogen or N2, consists of two nitrogen atoms, and they are held together by a triple covalent bond.
And a triple bond is, well, exactly what it sounds like, right? They're sharing 3 pairs of electrons.
Precisely. And in the world of chemistry, a triple bond is tremendously strong.
Like super glue strong.
Stronger. Those two nitrogen atoms are locked in this incredibly stable, highly unreactive death grip. They do not want to let go of each other to bond with anything else.
Wow.
So while this vital element is overwhelmingly abundant in the air, biologically speaking, it is entirely locked away behind a chemical vault.
Okay, let's unpack this. Imagine you are floating in a life raft in the middle of the ocean.
Okay.
And you are dying of thirst. There is water everywhere, in every direction, as far as the eye can see. But you can't drink it because it's too salty.
That's a great analogy.
Right. So atmospheric nitrogen is our ocean. It is everywhere, but it's completely inaccessible to our bodies. And nitrogen fixation, the process we're talking about today, that is the desalination plant.
Yeah.
It's the process of taking that unusable ocean and turning it into something that actually sustains life.
That is a perfect way to conceptualize it. And what's fascinating here is the sheer energy required to run that biological desalination plant.
Because breaking that bond is hard.
Oh, incredibly hard. Breaking a triple bond doesn't just happen on its own. It requires an immense amount of chemical force. But the biological demand for it is absolutely relentless.
Right. Life needs it.
Desperately. And we can see exactly how relentless through something called the Redfield ratio.
Oh, right. Discovered by the scientist Alfred Redfield.
Yes.
Yeah.
So back in 1958, Redfield analyzed the biomass of plankton in the ocean, and he found a remarkably consistent stoichiometric relationship.
A ratio of what they're made of.
Exactly. He discovered that for every 100 atoms of carbon that plankton assimilate to build their bodies, they require roughly 16 atoms of nitrogen
16.
and
Right.
1 atom of phosphorus.
So the ratio is essentially 106 to 16 to 1. That means for every single piece of biological growth, nitrogen is in massive demand.
Exactly. It is an absolute necessity for life to scale. Every time a cell divides, every time a strand of DNA is replicated, that nitrogen is required.
But again, the supply is locked behind this impenetrable triple bond.
Right.
So this is the ultimate evolutionary bottleneck. If the bond is that strong, and life desperately needs it to build DNA and proteins, nature had to figure out a way to break it.
It had no choice.
Which means life had to evolve a highly specialized, incredibly energy-intensive mechanism just to survive on this planet.
And it did. Biological nitrogen fixation, or BNF, relies entirely on a highly complex enzyme. It's called nitrogenase.
Nitrogenase, okay.
Yeah, this is the biological key that unlocks the nitrogen vault. It converts that stubborn atmospheric nitrogen into ammonia.
Which is the usable form.
Right, ammonia is what organisms can actually use to build biological structures, but it does not come cheap.
Yeah, the sources mention the cost in terms of ATP, and ATP is basically the cellular energy currency, right?
Correct. ATP is how cells pay for things to get done, and it takes 16 equivalents of ATP just to convert one single molecule of N2 into 2 molecules of ammonia.
16 ATP for one molecule. That's like paying 16 months of rent for a single cup of coffee.
It really is.
That is an outrageous energy bill for a microscopic organism to pay just to get raw materials.
It's a staggering metabolic cost, but the mechanism itself is an absolute marvel of evolution. Instead of trying to smash the triple bond all at once, the conversion happens at a very specific active site on the enzyme.
Like a specific tool.
Yeah, it's a metal cluster called FOMOCO, which stands for iron molybdenum cofactor.
And how exactly does that work? Because the sources mention protonation and reduction steps, but what does that actually look like inside the cell?
Think of the FOMOCO metal cluster like a microscopic crowbar.
A crowbar?
Yeah, it doesn't shatter the bond instantly. Instead, it sneaks hydrogen atoms into the triple bond one by one.
Oh, wow.
It pries those two stubborn nitrogen atoms apart, click by click, adding hydrogens until the triple bond finally snaps. And that separates the nitrogens and forms ammonia.
That is incredible. A microscopic crowbar operating on an atomic level.
Exactly.
And based on the evolutionary timelines in our sources, this specific enzyme, nitrogenase, evolved somewhere between 1.5 and 2.2 billion years ago.
Though some isotopic evidence suggests it could be as old as 3.2 billion years.
Which is wild. And what's interesting about that timeline is the metals involved.
Right, the iron and molybdenum.
Yeah, the exact form of nitrogenase, whether it relies on molybdenum or sometimes vanadium or just pure iron, was likely driven by whatever specific metals were abundant in the chemical soup of the ancient oceans.
At those exact moments in Earth's geologic history.
Right. Life literally used whatever tools it had lying around to build this crowbar.
It's highly opportunistic.
Okay, wait. I'm struggling with the logistics here.
Yeah.
You just described this enzyme as an evolutionary miracle that took billions of years to perfect.
It is.
But the sources also clearly state that nitrogenase has a fatal flaw. It is rapidly degraded and destroyed by oxygen.
Yeah. That is the great catch of biological nitrogen fixation.
But how is that possible? Wait, if this enzyme is destroyed by oxygen and is the only biological way to access nitrogen, how is it surviving out in the open?
It's a huge problem.
Because we live on a planet covered in oxygen-producing plants and breathing animals. Having an enzyme destroyed by oxygen seems like, I don't know, trying to build a matchstick house inside a blast furnace.
It really does seem impossible, but it's a brilliant biological puzzle. And nature came up with a series of incredibly intricate, localized workarounds to solve it.
Okay, like what?
Well, since many bacteria cease producing the enzyme the second oxygen is present, they had to adapt based on their environments. Some, like obligate anaerobic bacteria, simply live in environments entirely devoid of oxygen.
Oh, so just hiding from it.
Exactly.
Yeah.
Deep in the mud or at the bottom of the ocean. Problem solved.
Sure, but what about the bacteria that live right out in the open air or water?
That's where evolution gets highly creative. Take cyanobacteria, for example.
Okay.
These are photosynthetic bacteria. They inhabit almost all illuminated environments on Earth, and they actually produce oxygen themselves.
Wait, they make oxygen, but they need the enzyme that oxygen destroys.
Yes. So to protect their delicate nitrogenase from the very oxygen they are pumping out, species like Aphanizomenon and Dolichospermum develop specialized, thick-walled, oxygen-free cells. They're called heterocysts.
So they literally build a sealed panic room just to fix nitrogen.
Exactly. They create a microscopic panic room where the oxygen can't penetrate, and that is the only place where the nitrogenase crowbar goes to work.
That is wild. And what about plants? Because we constantly hear about farmers planting certain crops to put nitrogen back into the soil.
Well, technically, the plants don't fix it themselves.
Oh, they don't.
No, they form symbiotic relationships with the bacteria that possess the enzyme. The most famous examples of this are legumes. Think of crops like clover, soybeans, alfalfa, peanuts.
Kudzu.
Right, kudzu too. These plants form little root nodules that act as housing for rhizobia bacteria.
Okay, so they give the bacteria a home.
Yes. But to solve the oxygen problem inside those nodules, the plant does something amazing. It produces a specialized protein called legumoglobin.
Like hemoglobin. That sounds like hemoglobin.
It's structurally very similar to the hemoglobin running through your own blood right now.
Okay, so what does hemoglobin do inside a plant root?
It acts as an oxygen scavenger. It binds to the ambient oxygen inside the nodule, keeping it safely away from the sensitive nitrogenase enzyme,
Oh, wow.
but it still allows the bacteria just enough oxygen to respire and survive.
So the plant essentially puts the oxygen on a molecular leash. It keeps it perfectly suspended so the bacteria can breathe, but it can't touch the enzyme.
Precisely. It's an elegant solution, and this sort of biological hacking isn't just ancient history either.
No.
No, it's an ongoing evolutionary process. The sources highlight a mind-blowing recent discovery in a species of marine algae called Bermudosphaera bigelowii.
Oh, the nitroplast.
Yes. Scientists found an organelle inside this algae called a nitroplast, and it originated from a cyanobacterium known as UCYNA.
Let me make sure I'm wrapping my head around this. An organelle-like.
Yeah.
How mitochondria are organelles functioning as the powerhouse of our cells.
Exactly like that.
Wow.
What this implies is that a free-living nitrogen-fixing bacterium was at some point engulfed by the algae?
Like it ate it.
Basically. But over time, instead of being digested as food, it became a permanent, integrated part of the cell's machinery.
That's crazy.
Its sole dedicated purpose is now turning nitrogen gas into a biologically available form for the host cell. We were literally watching the evolution of a new cellular organelle happening right in front of us.
Okay, so nature's hackers, these deep mud bacteria, the microscopic panic rooms, the root nodules, they figured out how to make this desalination plant work.
They did.
But here's the massive structural problem that changed human history. Biological fixation is an incredibly slow, painstaking process.
Very slow.
A clover field can only fix so much nitrogen in a season.
That's right. It has a strict capacity limit.
And as human civilization developed and our population started to multiply, we hit a terrifying mathematical wall.
A huge wall.
We simply needed more nitrogen to grow our crops than the bacteria in the soil could biologically fix.
Without more fixed nitrogen, the carrying capacity of the Earth for human life was hard-capped. By the 19th century, the soil in Europe and North America was running out of nutrients.
It was exhausted.
Completely. We were facing a future where we simply could not grow enough food to feed the booming population.
Which means humanity had to look beyond biology. We had to figure out a way to break that triple bond ourselves.
Yes.
And ironically, before human chemists cracked the code, there was basically only one major non-biological way nitrogen ever got fixed on Earth.
Lightning.
Atmospheric fixation. It's a brilliant, violent alternative to the slow biological enzyme.
How does that work?
Well, when a lightning bolt strikes, the extreme localized heat of the plasma generates enough shear energy to violently rip those unyielding N2 triple bonds apart.
So the sheer temperature just overpowers the bond. Exactly. Once shattered, those freed nitrogen atoms immediately react with the oxygen in the air, forming nitrogen oxides.
Okay.
As those molecules cool down and interact with water vapor in the atmosphere, they eventually form nitrous or nitric acid, which then rains down into the soil as usable nitrates.
Here's where it gets really interesting, because when you look at the early human attempts to artificially fix nitrogen, those scientists were essentially trying to build machines to bottle lightning.
They really were.
Our sources mention Henry Cavendish way back in 1784.
Yes, Cavendish was one of the first to truly experiment with this.
He was literally using electric arcs to react nitrogen and oxygen in the air inside glass tubes. He was trying to mimic a thunderstorm in a laboratory.
It was very crude, but the concept was sound.
And that same underlying concept was eventually scaled up into the Birkeland-Eyde process in 1903.
But these early methods were incredibly inefficient. It was a desperate quest. Humanity was watching its population grow and realizing the agricultural collapse was imminent.
The pressure was on.
Huge pressure. The historical progression of these experiments reflects this escalating, almost frantic search for a chemical solution.
Right, because electricity wasn't cutting it. So they turned to brute force chemistry.
Exactly. In the 1860s, you had chemists Marguerite and Sarah Duval. They took barium carbonate and reacted it with nitrogen gas at intensely high temperatures to produce barium cyanide,
Oh, wow.
which they would then react with steam to finally get ammonia.
That sounds like a tremendously complicated workaround.
It was, and it was followed in 1898 by the Frank-Caro process, which created calcium cyanamide. Every iteration, every new chemical process was a desperate attempt to find an industrial way to pry that triple bond open and feed the dying crops.
But all of those electric arcs and barium experiments were just a warm-up. They were completely eclipsed by a single, brutal chemical process that fundamentally changed the trajectory of the human race forever.
You're talking about the Haber process, widely known as the Haber-Bosch process, discovered in 1909.
Yes, 1909. You want to talk about humanity brute-forcing nature? This is the ultimate example.
It really is.
To do exactly what tiny rhizobia bacteria do calmly at room temperature inside a delicate clover root, the Haber-Bosch process requires conditions that sound like they belong on the surface of Venus.
The conditions are remarkably violent.
We are talking about 200 atmospheres of pressure inside massive steel reactors. For context, that is the kind of crushing pressure you would experience if you were thousands of feet underwater at the bottom of the ocean.
Just immense pressure.
And it operates at temperatures of at least 400 degrees Celsius.
Yeah.
Which is hot enough to melt lead.
Yeah.
It uses massive amounts of natural gas as a source for hydrogen and literally pulls the air around us into the machine as a source for nitrogen.
It is the absolute definition of severe industrial catalysis. They use an iron-based catalyst, forcing the nitrogen and hydrogen gases together under that immense heat and pressure.
Until the bond just gives up.
Until the triple bond breaks and ammonia is formed. And the reality is...
It worked.
It worked perfectly.
It worked so well, it literally reshaped the planet.
Today, that single industrial method underpins the manufacture of virtually all nitrogenous industrial products globally.
That's crazy to think about.
We are talking about the foundation of modern synthetic fertilizers, which is its primary use. But the ammonia produced by Haber-Bosch is also the precursor for pharmaceuticals, textiles, commercial dyes, and, crucially, given the history of the 20th century, explosives and munitions.
So what does this all mean for us today? Our sources credit this exact process with supporting the rapid, unprecedented expansion of the human population.
Yes.
We went from roughly 2 billion people in the early 20th century to the 8 billion people alive right now.
That is the direct result of synthetic fertilizer.
Let me just make sure I am truly understanding the scale of this. Does this mean that out of the 8 billion people walking the earth today, roughly 6 billion of us literally only exist because of a violent chemical factory process invented in 1909?
If we connect this to the bigger picture, yeah, that is the stark mathematical reality.
Unbelievable.
Fertilizer production, via Haber-Bosch, is now the largest source of human-produced fixed nitrogen in the entire terrestrial ecosystem. Because we synthesized this process, we removed the biological speed limit.
We hacked the system.
We were able to grow food at a scale nature never, ever intended.
That is mind-bending.
Which means a massive, massive percentage of the nitrogen atoms currently sitting inside your own DNA, the structural backbone of the proteins making up your muscles, your skin, your heart.
Yeah.
At some point in the recent past, those exact atoms passed through a high-pressure, 400-degree steel reactor in an industrial chemical plant.
That is almost terrifying to think about. We are quite literally industrial products walking around in organic bodies.
We are completely tethered to the infrastructure we created. Right. Without the Haber-Bosch process running constantly across the globe, the global food supply would collapse almost overnight.
Wow.
We outgrew nature's desalination plant a century ago.
Okay, let's bring this all together. We started with a stubborn, locked-away gas that makes up almost 80% of our atmosphere. A molecule bound so tightly, it is basically useless to us in its natural form.
Right.
We saw how ancient biology fought an eons-long war, evolving highly sensitive enzymes like nitrogenase, utilizing microscopic crowbars to pry atoms apart.
And building literal panic rooms inside cells just to protect those enzymes from oxygen.
And then we saw how humanity, faced with mass starvation and a hard mathematical limit on our own growth, decided to bypass biology entirely.
We forced our way through.
We used extreme heat, incredible crushing pressure, and vast amounts of fossil fuels to mimic the shattering energy of lightning. We forced that molecule apart to feed the world.
It is an incredible journey. But the text does mention that scientists are actively struggling to find better ways to do this.
Right. There's a whole field called homogeneous catalysis, where researchers are desperately searching for low-energy catalysts to fix nitrogen.
They want to figure out how to do what tiny cyanobacteria do at room temperature.
But on an industrial scale. So we aren't so reliant on the massive energy demands of Haber-Bosch.
It is the holy grail of modern chemistry. Can we crack the code to artificially fix nitrogen as efficiently and elegantly as nature does?
Exactly. So the next time you sit down to eat a piece of bread or a bowl of rice or literally anything containing soybeans, remember that you are experiencing the end result of an evolutionary and industrial battle to break a single molecular bond.
Every bite you take is a tiny victory over the physics of the nitrogen atom.
Surviving only because somehow we learned to build the plant. But before we go, consider this one final thought. Because we are now artificially pumping millions of tons of this synthetic nitrogen into the earth every single year to grow our food, it isn't just staying neatly in our crops.
No, it definitely isn't.
It is bleeding into our soil, running off into our rivers, and flooding our oceans. We are fundamentally altering ancient ecosystems that haven't experienced this level of free nitrogen in billions of years.
It's a massive shift.
We solved the bottleneck of human survival, yes. But we might have accidentally started a brand new biological war in the process. Something to think about the next time you look at the ingredients on your plate. Thanks for diving deep with us today. Keep looking at the invisible world around you.